Usually when we think about a biological system, we sort of just assume science mapped it out perfectly centuries ago.
Oh, right, like it's just settled history.
Exactly. Take the circulatory system, right? The heart pumps blood, the veins carry it back. We've known the basic mechanics of that loop for hundreds of years. It feels completely, totally settled.
It really does. We tend to view most human anatomy as this solved puzzle, something structurally finalized by early Renaissance medicine.
But then you look into the female reproductive system, and suddenly that pristine textbook is just full of scribbles, crossouts, and pages that were literally glued together until, like, 20 years ago.
Yeah, it defies logic. How a physiological system that is universally relevant, one that every single human being relies on for their very existence, could remain so fundamentally misunderstood for such an incredibly long time.
Right? So welcome to this deep dive. If you're joining us, it's because you are insanely curious and you want the ultimate shortcut to being well-informed. Today, we are taking a massive stack of medical history texts, endocrinology journals, and recent anatomical studies to extract the real story behind this system.
Yeah, and it is quite a story.
It really is. We are going to map out the physical structures, decode the hormonal control room that runs them, and trace the history of how this vital biology was buried under centuries of just incredibly bad takes.
We're also going to look closely at the physical mechanisms. We want to move beyond just listing parts and actually uncover how the biochemistry dictates the physical anatomy, and why specific structural quirks have driven some of the biggest medical milestones in human history.
Okay, let's unpack this.
Okay.
Because I want to stress this isn't going to be some dry biology lecture. This is a story of a 2,000-year-long argument.
That's a great way to put it.
But to understand the argument, we really have to establish the physical baseline first. Clinically speaking, we are looking at two main divisions, correct?
Yeah. Structurally, yes. Medicine essentially divides the system into internal and external components. The internal organs tucked within the pelvis include the vagina, the cervix, the uterus, the fallopian tubes, and the ovaries.
Right.
And then the external genitalia are collectively categorized as the vulva. Additionally, the breasts are classified as accessory reproductive organs, given their functional role in nourishing offspring post-birth.
That sounds incredibly tidy and straightforward.
Yeah.
But diving into the historical texts, it becomes very clear that for the vast majority of Western medical history, none of those structures were actually described on their own terms.
No, not at all. They were viewed through a highly specific, very distorted lens. The historian Thomas Laqueur actually coined a term for this, the one-sex model.
And tracing that back, we land in the 2nd century with Galen of Pergamon.
Right, the guy whose writings basically became unshakable medical dogma for Europe for the next 1,400 years.
Yeah, his theory was that female anatomy wasn't a distinct system at all, but rather just an inverted, inferior male system.
The underlying logic for Galen and his contemporaries was based on these ancient theories of bodily humors and heat. The prevailing belief was that women simply lacked the necessary quote-unquote vital heat to push their genitals outward during development.
So they literally just thought the organs got stuck inside.
Basically, yeah.
Yeah.
So when early anatomists looked at the internal organs, they forced them into male categories. The ovaries were designated as female testicles. The vagina was described structurally as an internal, unextruded penis.
Which is just wild to think about. They weren't seeing two different biological blueprints. They were looking at a single male blueprint and categorizing the female version as just a colder, lesser state of it.
Exactly.
It's like early explorers mapping a brand new continent but stubbornly drawing it to look exactly like Europe because they literally couldn't comprehend that a different landmass could even exist.
The ideological bias completely overrode their physical observations. By the time we reach the Renaissance in the 1500s, you have brilliant anatomists like Andreas Vesalius conducting actual empirical human dissections.
Right. He's literally opening up the body and looking at the structures.
Yes. Yet, when he drew the uterus and vagina, he still illustrated them to visually mimic male anatomy. The cultural conditioning literally blinded them to what was sitting right in front of their scalpels.
And it took until the late 1700s and into the 1800s for European medicine to finally shift to a two-sex model, where the female system was recognized as categorically distinct and having its own unique physiology.
Which was a huge leap forward, obviously.
I think the mapping of the continent was finished then, right? But jumping into the modern anatomical studies in our source pile, it is mind-blowing to realize it took until the late 1990s, the era of dial-up internet and cell phones, to correctly map the clitoris.
That is easily one of the most stunning corrections in modern anatomical science. Between 1998 and 2005, an Australian urologist named Helen O'Connell published a series of MRI and anatomical studies that radically redescribed the structure.
Because before her, what did the textbooks say?
Prior to her work, standard medical texts, including major editions of Gray's Anatomy, depicted it merely as the small visible external nub, the glans.
Which showing only the glands is literally just showing the tip of the iceberg and totally ignoring the mountain of ice underneath the water.
Precisely. O'Connell's imaging demonstrated that the visible portion connects to a massive, mostly internal structure. It has these internal appendages called crura, or legs, and large vestibular bulbs that extend several centimeters deep into the pelvis, wrapping around the vaginal canal and the urethra.
It's a huge network.
It is. When you account for the entire hidden network, it is comparable in erectile tissue volume to the structures found in the penis. It was just systematically minimized or omitted from the literature for over 100 years. What's fascinating here is that the quote-unquote origin of this biological concept isn't a single flash of discovery. It is literally a 2,000-year argument.
Right.
Every successive generation of anatomists has had to physically correct the inherited ideological errors of the people who came before them to finally see the true structures.
Yeah. Now that we have a clear view of how the map was finally drawn accurately, we can move into how those structures actually operate, starting at the literal source. The ovaries.
Ah, yes, the ovaries.
Here's where it gets really interesting. Because the basic mathematical realities of the egg supply defy how we usually think about cellular biology.
We have to establish the concept of the finite egg bank here. Human biology approaches male and female reproductive cells with completely opposite strategies.
Right, because sperm are manufactured continuously millions a day throughout a man's lifetime.
Exactly. But oocytes, the egg cells, are produced entirely before birth.
I was reading through the endocrinology data on this, and the timeline is staggering. A female fetus at just 20 weeks of gestation, so she's still halfway through developing inside the womb, already possesses her absolute lifetime maximum of eggs. We are talking an estimated 6 to 7 million of them.
Right. And from that high water mark, the body immediately begins a process of massive cellular destruction. Through a natural, programmed cell death mechanism called atresia. That staggering 7 million drops to roughly 1 to 2 million by the day she is born.
And the attrition doesn't even stop there. By the time puberty hits, the reserve is down to about 300,000 to 400,000. And out of those hundreds of thousands, only about 300 to 400 will actually ever reach maturity and be ovulated in a single lifetime.
It's a massive reduction.
It's like having a trust fund where you start with $7 million, but the bank forcibly burns 99.9% of your cash before you're even allowed to make your first withdrawal.
That's a perfect analogy. It appears incredibly wasteful from an engineering standpoint. But in evolutionary biology, this intense selectivity ensures only the most viable, highest quality genetic material actually makes it to ovulation.
Although, looking at the recent literature, the absolute strictness of this finite bank has sparked some fierce academic battles. Which brings us to the Tilly controversy from the early 2000s, right?
Oh, yes. In 2004, Jonathan Tilly, a reproductive biologist, published a highly provocative paper in the journal Nature. He claimed to have found stem cells within adult mouse ovaries that were capable of generating brand new eggs throughout life.
Wait, if that translated to humans, it would completely rewrite the foundational rules of reproductive biology. It would mean the bank isn't actually finite.
Which is exactly why it triggered years of massive pushback, intense scrutiny, and multiple failed replication attempts by other labs. Today, the concept remains highly contested. Mainstream reproductive endocrinology still operates on the foundational principle that the human ovarian reserve is strictly non-renewable.
So assuming this shrinking vault of eggs is the reality, the ovaries aren't just sitting there passively storing them, they actually act as the physical hardware for an incredibly complex brain-controlled command center called the HPO axis.
The hypothalamic pituitary ovarian axis. This biochemical feedback loop dictates the entire cycle. It demonstrates how hormones coordinate physical changes across vastly different parts of the body.
Let's break down the mechanics of that loop. Because when you hear terms like axis, it can sound like abstract machinery, but this is a very physical chain reaction starting deep inside the brain.
The cascade begins in the hypothalamus, a small region at the base of the brain. It secretes a hormone called GnRH in steady, rhythmic pulses. Those pulses travel a very short distance down to the pituitary gland.
Okay, so brain to pituitary.
Right. And the pituitary responds by releasing two distinct hormones into the main bloodstream, FSH and LH.
Let's ground those acronyms real quick. FSH is follicle-stimulating hormone, which does exactly what it says on the tin. It travels through the blood, down to the ovaries, and stimulates a group of follicles, the little sacs holding the eggs, to wake up and start growing.
Exactly. And as those follicles grow, they begin pumping out estrogen. Here, we encounter a brilliant first principles lesson in endocrinology. Initially, as estrogen levels rise in the blood, it exerts a negative feedback loop on the brain. It signals the pituitary gland to slow down the production of FSH.
A lot of textbooks compare that to a thermostat. Heat rises, so the furnace turns off. But that analogy doesn't really capture the full mechanism here, because estrogen doesn't just turn the system off.
No, it does something much more dramatic.
Right. It's more like water building up behind a dam. At first, the rising water tells the operators to slow the river flow. But once that estrogen reaches a critical overflowing height and stays high for a few days, it triggers an emergency protocol. The negative feedback suddenly flips to positive feedback.
That is a much more accurate visualization. The sustained high estrogen forces the dam to burst, which causes the pituitary gland to release a massive sudden surge of LH luteinizing hormone.
And that's the trigger.
Yes. That sharp chemical spike travels to the ovary and causes the dominant follicle to physically rupture, releasing the mature egg. That localized physical rupture is ovulation.
The exact same hormone, estrogen, has completely opposite effects on its own control center, just depending on its concentration and the timeline. It suppresses the signal to keep things orderly and then triggers an explosion when it's time to act.
It illustrates perfectly how dynamic and deeply time-sensitive human biochemistry really is.
So, with ovulation triggered by the LH surge, the egg is physically ejected from the ovary. But tracing its subsequent physical journey through the anatomy reveals some structural designs that are honestly a bit baffling.
We definitely have to address the gap.
Yes, the structural gap between the ovary and the fallopian tube. Because logic would suggest the tube should be a sealed pipe attached directly to the ovary to safely transport the egg. But they don't actually touch.
They don't. There is a literal physical space in the abdominal cavity between the surface of the ovary and the fringed funnel-like end of the fallopian tube, known as the infundibulum.
Having the egg just eject into an open space seems like dropping a vital microscopic package over a chasm and just hoping the recipient catches it on the other side. From an evolutionary perspective, how does that open gap ensure the egg doesn't just float away into the abdomen?
Well, it relies on fluid dynamics rather than a sealed physical connection. The fringed ends of the tube, called fimbriae, become highly active around ovulation. They are lined with microscopic hair-like structures called cilia.
Like little brooms.
Kind of, yeah. These cilia beat rhythmically, creating a literal fluid vacuum, a sweeping current that pulls the peritoneal fluid and the egg along with it across the gap and into the tube. It really highlights how evolutionary biology often settles on mechanisms that are good enough rather than perfectly engineered.
But because it relies on a fluid current rather than a closed pipe, things can occasionally go wrong.
Unfortunately, yes. The open anatomy is the exact reason why ectopic pregnancies can sometimes implant outside the reproductive organs, in the abdominal cavity. The egg was never inside a fully sealed transit system to begin with.
Wow. Okay, so once the egg is successfully vacuumed into the fallopian tube, there's another major misconception to clear up. If you ask most people where a sperm fertilizes an egg, they picture it happening inside the main cavity of the uterus.
Which is almost never the case. Fertilization almost never happens in the uterus. The meeting point is the ampulla, which is the widened middle section of the fallopian tube.
And there's a real time crunch there.
Right. A severe one. The egg only remains viable for about 12 to 24 hours after ovulation. The sperm must navigate all the way up into the tube to meet it within that incredibly tight window.
If that fertilization is successful, the newly formed embryo still has a 3 to 5 day physical journey down the remainder of the tube before it even reaches the uterus to implant. And while that multi-day transit is happening, the ovary isn't just taking a break, it's actively keeping the future environment viable.
Right, the ovary performs a remarkable piece of physiological remodeling. The ruptured follicle that just released the egg doesn't just heal over, it chemically transforms into a brand new temporary endocrine gland called the corpus luteum.
It builds a hormone factory on the fly.
Exactly. This temporary factory starts pumping out massive amounts of progesterone. Progesterone is the hormone that stabilizes and enriches the uterine lining, preventing it from shedding. The corpus luteum acts as a critical physiological bridge, sustaining the pregnancy for the first 8 to 10 weeks until the placenta can fully develop and take over hormone production.
Okay, so while the fallopian tube and the ovary are handling the egg, we have to look at the entrance to the system from the other direction. The cervix, which sits at the base of the uterus. For a long time, the cervix was viewed as just a passive anatomical doorway.
Which completely ignores its complex cellular function. The cervix is highly active. It functions as a programmable gatekeeper. The cells lining the cervical canal produce mucus, and the physical architecture of that mucus changes radically in response to the brain's hormonal loop.
So for the vast majority of the cycle, when estrogen is low and progesterone is dominant, the cervix creates a dense, highly acidic mucus. If you look at it under a microscope, it basically forms a dense briar patch that is totally hostile to sperm.
Right, it acts as a physical and chemical barrier to keep the sterile environment of the uterus safe from external bacteria.
But as that estrogen dam builds up right before ovulation, the cervix receives the signal to alter the mucus production. It becomes thin, highly alkaline, and stretchy, what clinicians often describe as an egg-white texture.
Yeah, structurally, the microscopic mesh actually aligns into parallel channels. It actively builds a biochemical highway that protects sperm from the acidic vagina and guides them up into the uterus.
It literally dismantles the barricade, builds a specialized transit route for a few days, and then the moment ovulation passes and progesterone rises, it rebuilds the wall.
Grasping that cyclical structural change to the mucus is the entire physiological basis for the Billings ovulation method, developed by Australian physicians John and Evelyn Billings in the 1950s and 60s as a fertility awareness protocol.
So having traced this entire sequence, from the ancient anatomical errors to the HPO axis, across the gap and through the cervical gatekeeper, we really need to zoom out. Because treating this system as a mystery box hasn't just resulted in bad textbook drawings. It has generated deep-seated societal myths, and overcoming those myths has driven some of our biggest medical breakthroughs.
Oh, absolutely. The lack of accurate anatomical education still fuels basic misconceptions today. I mean, if you've ever downloaded a cycle tracking app on your smartphone, you've probably noticed it aggressively defaults to predicting ovulation on day 14 of your cycle.
Which assumes that every single human operates on a flawless, factory-standard 28-day schedule.
That day 14 default is a massive oversimplification. The cycle is divided into two parts. The pre-ovulation phase, the follicular phase where the follicles are growing, can vary wildly. For some, it takes 10 days, for others 20.
Ah, so that part fluctuates.
Yes, it's only the post-ovulation luteal phase, governed by the lifespan of the corpus luteum, that is a relatively fixed 12 to 14 days.
Another physical myth that circulates heavily is the idea that the ovaries take polite turns, strictly alternating left and right every month.
Which is totally false. Which ovary releases an egg in any given cycle is essentially random. Both ovaries receive the FSH signal from the brain, and multiple follicles may start developing on both sides. Ultimately, a complex local biochemical race dictates which single follicle becomes dominant and suppresses the rest.
We also see ancient myths about the physical nature of menstruation still lingering. For centuries, cultural texts framed menstrual blood as a toxic purge, a way for the body to rid itself of poisons.
Physiologically, menstrual fluid is simply the controlled shedding of the endometrial tissue and blood that was built up to support a potential pregnancy. It carries no unique toxins or poisons. Framing a standard physiological reset as a purge has historically served to rationalize intense menstrual stigma rather than reflect biology.
And the history of weaponizing this anatomy goes back to Hippocrates in the 5th century BCE. He documented a massive range of symptoms, anxiety, breathlessness, seizures, and attributed them all to a wandering womb.
The hypothesis was that if the uterus was deprived of childbearing, it would become starved and literally detach, migrating throughout the body cavity to press on the lungs or brain, causing physical and mental illness.
Which is anatomically impossible, given the heavy ligaments that anchor the uterus deeply into the pelvis.
Completely impossible.
But the logic was so culturally accepted that it gave us the enduring psychiatric diagnosis of hysteria, a word derived directly from hystera, the Greek word for uterus.
It is the ultimate textbook case of how a purely structural misunderstanding of an organ can dictate centuries of medical and social mistreatment.
As modern medicine began to finally map the real mechanics, the discoveries connected to this system led to world-changing milestones. The understanding of the cells in this system gave us one of the most vital research tools in existence, HeLa cells.
Yes. In 1951, Henrietta Lacks underwent treatment for an aggressive cervical cancer at Johns Hopkins. The biopsy of her cervical tumor yielded cells that behaved unlike any human tissue previously observed.
It just kept dividing, right?
Exactly. They didn't die after a few divisions. They became the first human cell line to survive and multiply indefinitely in a laboratory setting.
The biological resilience of those specific cervical cells underpinned the development of the polio vaccine, foundational research into HPV, and vital work in genetics. The female reproductive tract provided the physical engine for modern virology.
And our mastery of the fallopian tube's exact environment led directly to in vitro fertilization. In 1978, physiologist Robert Edwards and gynecologist Patrick Steptoe oversaw the birth of Louise Brown, the first IVF baby.
To achieve that, they essentially had to act as the HPO axis and the ampulla. They used hormones to stimulate the ovaries, extracted the egg right before the dam burst, fertilized it in a dish, and crucially, they had to replicate that 3 to 5 day transit time outside the body, waiting for the embryo to reach the right developmental stage before placing it into the uterus.
It's an incredible feat. But conversely, failing to respect the anatomy has caused devastating medical disasters. The Dalkin shield in the 1970s is a prime example of structural ignorance.
This was the incredibly popular intrauterine device, the IUD, that ended up causing widespread pelvic inflammatory disease. The catastrophe wasn't just a random side effect, it was a direct failure to understand the cervical gatekeeper we discussed earlier.
Right. The device featured a multi-filament string meant to hang through the cervix for removal. But structurally, that twisted string acted like a wick.
Oh, wow.
Yeah, through capillary action, it drew fluid and bacteria from the non-sterile vagina straight past the highly acidic, protective cervical mucus and deposited it into the sterile uterus. It physically bypassed the body's natural defense mechanisms.
That structural design flaw caused severe infections and multiple deaths, ultimately shaping modern medical device regulations and leaving a generational distrust of IUDs, even though modern designs use monofilament strings that don't wick bacteria.
The history of medical engineering requires a flawless understanding of the underlying biology, or the consequences are immediate and severe.
Before we wrap up the milestones, the evolutionary biology surrounding the end of this system's life cycle is just as fascinating as its beginning. The concept of menopause, the permanent cessation of ovarian function, is actually a profound anomaly in the animal kingdom.
Aside from humans, menopause is documented in almost no other terrestrial species. We share this trait primarily with a select group of toothed whales, specifically orcas and belugas.
Just humans and whales.
Mostly, yes. In most mammals, reproductive capacity continues until the end of the organism's lifespan.
This evolutionary puzzle led anthropologist Kristen Hawkes in the 1990s to champion the grandmother hypothesis. The theory proposes that human menopause evolved as a massive survival advantage. Post-reproductive women who shifted their energy away from the physical risks of bearing their own children and instead helped provision and care for their grandchildren actually ensured a higher survival rate for their shared genetics.
It's a fascinating way to reframe the end of the reproductive cycle.
So what does this all mean? We've covered a massive amount of ground, from Galen's inverted male anatomy and Vesalius's biased maps, to the trust fund of finite eggs, the dam-bursting precision of the HPO axis, and the evolutionary anomaly of orcas. When you look at the sheer scale of real-world impact, it's staggering. Consider conditions like endometriosis, where uterine-like tissue grows outside the uterus. It affects roughly 1 in 10 women globally, yet for decades the pain was structurally dismissed by clinicians. How should you, the listener, take all this physiology and apply it today?
If we connect this to the bigger picture, it becomes very clear. You simply cannot navigate modern medicine or even public policy without a firm grasp of this underlying anatomy. If you want to comprehend the debates surrounding fertility treatments, IVF regulations, and egg freezing, you have to understand the unyielding biology of the finite oocyte supply.
Right.
If you want to understand the monumental importance of the HPV vaccine, you have to look at the cellular history of cervical cancer and HeLa cells.
The same goes for understanding hormone replacement therapy studies, or movements addressing menstrual equity and period poverty.
Absolutely. None of those modern debates make logical sense if the foundational endocrinology and physical structures are treated as a mysterious black box. Understanding the system isn't just about passing a biology test. It is the fundamental prerequisite for informed healthcare, self-advocacy, and policymaking.
Thank you for joining us on this exploration of a system that is as dynamically complex as it is vital. We started with ancient philosophical errors and traced our way through the incredible biochemical precision of the HPO loop, the fluid dynamics, the fallopian gap, and the programmable barricade of the cervix.
I want to leave you with one final lingering thought to mull over. We sit here today looking back at the ancient Greeks, and we easily laugh at the absurdity of believing that a starved uterus wandered around the chest cavity, causing breathlessness.
We feel very scientifically superior.
We do. But remember the timeline we discussed? It took until the late 1990s and early 2000s, an era when humanity was already successfully operating remote-controlled rovers on the surface of Mars to accurately map the basic physical anatomy of the clitoris.
Putting the Mars rover on the exact same timeline as basic human pelvic anatomy is just a staggering reality check.
It really makes you wonder, with all our advanced imaging and supposed scientific objectivity, what other fundamental microscopic or structural realities in our own human bodies are we currently completely blind to? What is sitting right in front of our scalpels today, just waiting for the next generation's anatomists to finally strip away our modern biases, open their eyes, and actually look?
A perfect question to leave on. Keep questioning what you think you know, and we will catch you next time.